Hello everyone. In this session, we are going to discuss about the nucleic acid, that is RNA, which comes under the chapter Nucleic Acids of the paper 5, that is Cell Biology and Biotechnology. So in our previous classes, we have already discussed about the one more nucleic acid, that is DNA, and how the DNA is also proved it is also nucleic acid or a genetic material. Then also we have studied how DNA replicates. So now let's begin with what is RNA and the properties, the function and the types and their functions also. So we all are aware about this RNA also. It is also a type of genetic material, or it is also a type of nucleic acid which is single stranded when compared to DNA. But the RNA, how it will be formed? So once the DNA is undergoing transcription, then only RNA will be formed. So that transcription will be dealing out in later. So let's see what is RNA. So RNA is nothing but it is a ribonucleic acid which is made up of a polymer of nucleotides, and it contains ribose sugar. So the ribose sugar, phosphate and the bases. So bases are adenine, guanine, cytosine and uracil. Here the thymine, the nucleic acid the base of the DNA is replaced by the uracil. Thymine will be replaced by uracil. And this RNA is a polymeric molecule which is very much essential for coding and decoding, regulation and expression of genes also. So RNA is very important for transcription or the protein synthesis. So this is how the structure looks like, and the chemical composition of RNA, as I said, it contains a pentose sugar, that is RNA will be a ribose sugar. Nitrogenous bases, they are of two types, that is purines and pyrimidines. Purine will be adenine and guanine. Pyrimidine will be uracil and cytosine, because thymine is replaced by uracil. And phosphoric acid, which is very much responsible for giving acidic property to the RNA. And also if you see the nucleosides and nucleotides, so nucleosides are riboadenosine. How in DNA you had deoxyadenosine, here riboadenosine, riboguanosine, ribocytidine, ribouridine. And nucleotides will be adenylic acid or adenosine monophosphate, AMP, guanylic acid or guanosine monophosphate, GMP, cytidinic acid or cytidine monophosphate, CTD, or uridine monophosphate or uridylic acid, UTP. So these are the chemical compositions which are related to the chemical structures of RNA. So same like DNA, even RNA, it is a long polymer and it is a single stranded. And if you see the structure of DNA is somewhat same, but it is single stranded. The components are same. It is also having 5' end with the phosphate group and 3' end with the hydroxyl group. And they are composed of ribonucleotides. So till now what I said, nucleosides and nucleotides, so they are composed or they are arranged with the ribose sugar, hence they are called as ribonucleotides. And these ribonucleotides are linked by 3' to 5' along with the phosphodiester linkage. So phosphodiester linkage is very important for giving the structure, and nitrogenous bases, as I said, adenine, cytosine, uracil and guanine. And the difference in structure of RNA, that is the bases, because the thymine is replaced by uracil in RNA, and thymine uracil can form bases with the adenine. And sugar is ribose, and I already mentioned about the nucleosides, that is ribonucleoside, adenosine, guanosine, cytidine, uridine, and along with the nucleotides. So now the most RNA molecules are single stranded and can form complementary base pair with the RNA. So due to that, or they loop back, they are having different structures. So for that different structure, they can form double stranded in the form of TRNA, transverse RNA, mRNA, messenger RNA, then rRNA, ribosomal RNA. So these structures are very important, and they are very important for your examination point of view also. So as I said, there are the three different types: rRNA, tRNA, mRNA, ribosomal RNA, transfer RNA and mRNA, messenger RNA. So let's begin with the mRNA structure. As you can see in the picture, the mRNA, it looks like a linear strand and it has got two ends, namely 5' end and 3' end. And at the 5' end, you can see the guanosine. There you can see G and CH3, they are attached. So that region is called as 7-methylated guanosine. It is also called as cap. And this cap is followed by 50 to 100 non-translatable nitrogen base sequence. So that is called as leader sequence. If they are non-translatable, then they are called as leader sequence. And behind the leader sequence, there you can find the initiation codon or initiator codon, AUG, or you can find the GUG in prokaryotes. And after initiator codon, okay, what is the function? The initiator codon followed by the axons and later introns. But near to the 3' region, you can find the terminator codon also. So that will be untranslatable in nature, and this terminator codon, to that region there will be polyadenylic tail which is attached to the mRNA, so which is around 200 nucleotides longer. And in prokaryotes, the mRNA is polycistronic. So polycistronic means what now? They are having maximum or many cistrons, poly, which are attached to the mRNA. And whereas in eukaryote, it will be monocistronic, single. So poly A tail. So remember, this is the structure of mRNA, how it looks like. So this is the, and okay, only 5% of RNA will be having this mRNA, the messenger RNA. And what is the function? This mRNA carries genetic information from nucleus to cytoplasm and also helps in the protein synthesis. So this is about the structure of mRNA. So next, coming to rRNA. So rRNA is nothing but the ribosomal RNA. So this ribosomal RNA structure, if you see, it is a single stranded, and this ribosomal RNA, it represents around 80% of the RNA, and there are two major components, smaller subunit and larger subunit. In eukaryotes, it is synthesized on DNA by RNA polymerase 1. So you have studied the RNA polymerase enzyme in DNA replication. mechanism. So this RNA polymerase will synthesize the DNA. On the DNA in eukaryotes. So as I was telling, there are two subunits. ATS type you can find out in the eukaryotes. 70S type you can find in prokaryotes. So what is the function then? So function, it is helping in binding the ribosomes to the mRNA. It also holds the subunits of the ribosomes together. So two subunits are there. And it also acts as the enzyme, that is ribozyme. Helps in the formation of peptide bond formation during protein synthesis. So this is about the rRNA. So next coming to tRNA. So very important structure, can be asked for 7 marks. So all these structures are important: mRNA, rRNA and tRNA. So now when you see the structure of tRNA, so you can find there are different arms and it also looks like a clover leaf. So it is also called as clover leaf model. So this clover leaf model of tRNA, so it was given by Robert Hawley. So this is how it looks. So Robert Hawley, and while he was studying in East actually in 1965, and it resembles trifloriate leaf. So that is how the name comes, clover leaf. And you can find there are 4 arms: amino acid binding arm, or it is also called as acceptor arm. Then you are having the T5C arm or pseudouridine arm. Next, DHU arm, or you can call it as Dloop or dihydrouridine arm, and later you are having anticodon arm. These are the 4 important arms, and one more arm you can find, that is variable arm or variable loop. So what is the function of each loop now? So now each arm has having the stem and loop. So you can find, so branching is there, and along with that you can find loop formation is also present. So these each loop and stem, they are paired by nitrogenous bases and they are unpaired by the nitrogenous bases. Stem is paired and loops are unpaired. So as you can see, the stem region, they are paired by these nitrogenous bases. But when you see the loops, they are unpaired. So here also they are unpaired, here also they are unpaired. So loops are unpaired. And coming to the first, acceptor arm. So acceptor arm is nothing but the amino acid binding arm. So it has got 7 base pairs and 3 unpaired bases. And this amino acid is always bind to adenine present. So that is how you can call it as adenine or adenine binding amino acid arm or amino acid binding arm. Next, coming to T5C arm. So T5C arm has 5 base pairs and 7 unpaired base nitrogenous bases, and it also helps in binding tRNA complex, transverse RNA complex, at the time of protein synthesis. So T5C arm, it has got 5 base pairs and 7 unpaired bases, which helps in the protein synthesis. Next, anticodon arm. So anticodon arm, it has got 5 base pairs and 7 unpaired bases. Loop of the anticodon, what it is having? The nitrogenous bases are very much complementary to axons. And last, that is DHU arm or D5. Here the 4 bases are paired and 10 pairs are unpaired, and it is very much helpful in recognition of specific amino acid and activation of amino acid. So these are the functions of these different arms now. So this tRNA was discovered by Mohalon, and the structure was very much proposed by Kim in East structure. So this is how the tRNA, when it is made to see under tridimensional, that is 3-dimensional, and very much simplified single structure. So it has got the adapter molecules also, how they are very much, and this structure has got around 75 to 95 nucleotides totally. So, and the function is nothing but it mainly helps in protein synthesis, and it also transfers amino acids and corresponds 3 nucleotide codon, that is rRNA, to make polynucleotides and proteins. So to summarize the difference between DNA and RNA, and also you should know the properties of RNA before that. They also form nucleotides, so you know the properties and the characteristics, so you can write about this. So make a note. And they are more resistant to damage of UV light also than DNA. So DNA is very much, it can be easily damaged, but these are having resistant. And functions have mentioned in each among them of tRNA, mRNA and rRNA, that you can write during that. Okay, coming to the conclusion, the DNA, it is made up of deoxyribose, here ribose sugar, monomers, deoxynucleotides, ribose nucleotides, pentose sugar will be deoxyribose and ribose sugar, and nitrogenous bases are different because thymine will be replaced by uracil in RNA, and number of strands, usually DNA is double stranded and RNA is single stranded. Location of DNA synthesis will takes place in nucleus, whereas RNA is in cytoplasm. So that's all about today's session. So in this session we have discussed about what is RNA and types of RNA, functions. So very important for your examination point of view. So maximum tRNA structure they have asked many times. So that's about this session. Thank you.